Welcome back, and our next speaker is Professor Douglas Erwin. He's a paleobiologist and evolutionary biologist who studies large-scale patterns in history of life. He's famous for his work on Permian extinction, the biggest extinction in the past 600 million years, and probably the biggest in history of life. Well, considering that some argue that we have mass extinction going on right now, his work is highly relevant. And he just told me that we actually know now about the Permian mass extinction even less than we used to know 10 years ago and 20 years ago. So it's very, very interesting work. But some of his recent research also focuses on novelty and innovation in biological, cultural, and technological systems, and whether there is a conceptual framework that can encompass all three of these parts. So let us welcome on the stage Douglas Erwin.
Thank you. It's a pleasure to be here. I'd like to thank Jan for inviting me back. I was here at one of these, I think that one, quite a while ago. It's a pleasure to be back in Singapore. I want to talk about time from the standpoint of a geologist or a paleobiologist rather than a physicist or economist or some of the other speakers. Geologists are sort of parochial. We think we invented deep time. This is the front piece of a book by Thomas Burnet from 1684 called The Theory of the Earth, which was originally published in Latin and then published in English. And this is from the English version, which is the Sacred Theory of the Earth. And it looks like this is a cyclical theory with God on top and the sun and earth going through various stages here. In fact, this was the first real appreciation that the earth must have a much greater temporal dimension than most of natural theologians at the time, science didn't really exist, were willing to give it. Geologists tend to credit these two people, James Hutton from Scotland and then Charles Lyell, as being the founders of modern geology. Hutton famously said in his theory of the Earth that time has no vestige of a beginning nor prospect of an end, which most people have interpreted as there being this infinitely long history of the Earth, certainly a much longer history than most people in Western religions would give it at the time that Hutton wrote in the late 1700s. And Charles Lyell, Hutton basically was unreadable. You can read like a page or a page and a half of it and your eyes glaze over. And even in a Scottish brogue, it wouldn't have improved. So a man named Playfair, a friend of his, translated it from Huttonese into English. And then Sir Charles Lyell, in the 1830s, wrote a more comprehensive look at geology that was based upon a principle that became known as uniformitarianism. And the Principles of Geology was what the book that Darwin read as he was going around the world on the Beagle in the early 1830s. And he, in a letter to Sir Roderick Murchison, says, this will give an abstract of all that is known in geology. My view of these principles, no causes whatever have from the earliest time to which we can look back to the present ever acted but those which are now acting, and that they never acted with different degrees of energy from that which they now exert. This is the basic principle of uniformitarianism that is still taught for some inexplicable reason to geology students today. The present is the key to the past. By studying processes today, the Mississippi River or the formation of reefs in Australia or whatever, we can understand all of geologic history. And it's true that processes that we study today are useful, but the thing that Lyell stuck in here at the end is and that they never acted with different degrees of energy from that which they now exert. So he's making a substantive claim in this letter to Murchison about the rate, the activity of these different processes. And the problem with uniformitarianism, which has been key to geology since Lyell wrote this, is that the only people who believed this, even in 1829, were Charles Lyell, James Hutton, and Charles Darwin. Even at the time, nobody believed Lyell, because they knew that geological processes varied wildly in their rates over time. But Lyell actually got people to sort of believe this, at least into the 1960s or 1970s. And it's actually had a fairly pernicious impact on how we look at processes that are going on. I'll come back to this at the very end of my talk. This is a review of Lyell's book by Hewell, who criticizes Lyell for this very issue about the intensity of various geological processes. It wasn't until 1913 that we actually had the first radioactive or reliably dated geological timescale. During the 19th century, Darwin needed a lot of time for evolution to work in the origin of species. He has a long section in chapter 9, I think, of the first edition, in which he talks about the erosion and the denudation of the Weald in southern England as being one way of estimating how much time has elapsed since the Cretaceous. He was wildly off in how much time there was. And it's not until we get radioactivity that we actually have some way of establishing how much time we actually have for geological and also biological processes to operate on. Today, this is actually the very most latest version from the International Commission on Stratigraphy. We have a very well-dated timescale in many places. The sections that I work on at the Permo-Triassic boundary in China, which are, where's the Permo-Triassic? Right here. So we have this boundary here dated down to plus or minus 30,000 years ago. So we can, I'll show you this data a little bit later. We can actually discriminate in ideal sections down to tens of thousands of years, even for events that are 250 million years ago. That takes a phenomenal amount of work, mostly by my colleague Sam Bowring in his lab at MIT. But by dating radiometric ash dates, we can get lots of dates through here and get a very good idea of rates and other sorts of things through deep time. Now, what I want to do today is talk first very briefly about some of the complications of measuring rates, because that turns out to be really critical for a lot of what we want to understand as evolutionary biologists. I want to talk about tempos of evolution in the plural, making the point that there are many different tempos over which evolution operates, and that actually understanding the tempo of evolution is really dependent upon the question that you're trying to answer. One of the main points I'll make in this section is that evolutionary activities are often decoupled at different timescales. So the evolutionary events that happen within a species are often decoupled from those between species or those on longer timescales. Then I'll turn to the main section, in which I'm going to argue that there are a number of evolutionary novelties and adaptations that have happened that basically operate to modify the timescales on which organisms or on which evolution can operate within organisms. They do that by, the part of this that I'll emphasize is that they do this by generating what have been called slow variables. So they preserve stability in many biological processes from the pace of temporal change that evolutionary biologists believed even into the 1970s. So here's the first sentence of a book by Gregory Cochrane and an anthropologist named Henry Hardpenning, called the 10,000-year explosion. And how many people, particularly after what Jeffrey just said, believe this statement to be true? Show of hands, please. This is audience participation. How many people don't believe it? Okay. These people know me because they've seen it before. They don't count. So what's wrong with this statement? What's wrong with it is that this is true for any 10,000-year period of time, because what they've done is ignore the denominator. This happens more frequently than one would like to have happen when geologists and evolution biologists talk about rates. He's made a comparison of a short-term rate to a very long-term rate. If I look at the accumulation rates of siliciclastic sediments, so how much sand is going down a river and being deposited on the continental shelf, there's an inverse relationship here between the accumulation rate and the time over which this is measured. And it turns out if I measure rates of all sorts of things, I get exactly the same relationship. And the reason for this is that many of these processes vary over time. This is the flow of the Colorado River, estimated from tree-ring data from 1500 up until about 1900, and then measured by the US Geological Survey with the gray line that you may be able to see here. That's from a gauging station at Leesford, Arizona. And you can see that there's huge variations in the flow of the Colorado River, not only from year to year, but from month to month. Parenthetically, I will say that one of the reasons why the river is in such trouble is that when they built, the Bureau of Reclamation built dams along the river in the 1920s, they measured the flow rates right here. So at the highest flow rate since, well, almost the highest flow rate since the 1500s. And they based the estimate of the amount of water that Phoenix and Las Vegas and other places could take out of the river on water that basically didn't exist, which is why the reservoirs are drying up. This kind of thing happens not only in geology, but also in biology. So this is rates of viral evolution over time. These are different groups of viruses, and in all cases, there are these inverse relationships. And the reason this happens is because of something called mutational saturation. So unlike the case in the rivers, there's still varying rates over time, but certain sites can get saturated with different changes. In the case of a DNA sequence, so this is a genome, in this case, these are 2 yeast strains here with a number of sequences. There's only 4 different options for each particular nucleotide site. Once those changes have happened, all you can do is produce another T, A, C, or G. That kind of saturation also happens when we're looking at morphology. This is a study done by Phil Gingrich in 2000, looking at inverse rates of changes using experimental data, microevolutionary rates, historical data, and then long-term paleontological rates, showing this inverse relationship over time. It turns out that the reason for this in morphology, looking at horses or elephants or gastropods, snails, is the same as it is for viruses. There's a limited number of things that can change, and eventually, if you wait long enough, you've changed everything that you can change. So this relationship begins to flatten out with time. And this is a whole pile of data from a paper by my former PhD student, Pete Wagner, looking at everything from cardidid bivalves to trilobites, crinoids, horses. Sort of didn't matter what he looked at. They all show the same relationship. So when we get to tempos of evolution, this fundamental question that George Gaylord Simpson asked in 1944, how fast, as a matter of fact, do animals evolve in nature? It's a fundamental observational problem of tempo and evolution. The first line of his famous book, Tempo and Mode in Evolution, there's no single answer to this, and there can't be, because it depends upon the interval of time over which we're looking and the problem we're trying to solve. So these tempos, in the plural, have been a problem that paleontologists and evolutionary biologists have been studying really since Simpson pointed out the importance of these in 1944. I think we've identified some of the scope of the problem and realized that there is no single solution to the problem. And what I'm going to do now is take you through some of those different tempos that exist. And then I want to talk about how evolution has dealt with the problem of time in the sense of constraining the option space, the possibility space in which different types of evolution can operate. So if I was a population geneticist, I actually took population genetics. I did fairly well, so I can fight with those guys. The argument I would make is that evolution is about nucleotide changes on DNA, that all DNA is basically kind of the same. We have a robust mathematical theory in population genetics to tell us about the kinds of evolutionary change, and that will tell us about the evolution of cichlid fish on the top of this diagram, or different kinds of plants in Hawaii, or any scope of evolutionary change. And the kinds of evolutionary change that we examine in the laboratory or by field experiments today are sufficient to explain, by extrapolation, all of evolutionary change through time. By studying flies in bottles today, that will tell me, ultimately, what happened in the Cambrian 540 million years ago with the explosion of a whole variety of different kinds of animals. I'm not a population geneticist, so I'm not going to tell you that, because it's not right. I don't think it's right. The problem is that evolutionary change appears to be decoupled at over a number of different temporal spans through the history of life. The first of these is what was described as punctuated equilibrium by Niles Eldredge and Stephen Jay Gould in the 70s. This is an example by a colleague of mine, Alan Cheatham, looking at a bryozoan called Metrorhabdotis from Panama. Bryozoans are colonial organisms, so each of the holes in this SEM photo here had a different individual living in it, but they all form a big colony. And Alan and Jeremy Jackson looked at these bryozoans through about 20 million years of history of the fossil record in Panama, but because several of these were living today, and they also looked at a different genus that has more living specimens, they were able to relate the taxa based on the fossils to the genetics of the taxa as well. And they identified stability within many of these species lineages with abrupt transitions to new species. This pattern of punctuation that Steve Gould described in the early 1970s. More recently, people who do this sort of work, particularly my colleague Gene Hine at the museum, have spent a lot of time collecting a large number of studies to examine whether they fall into predominantly into stasis, a random walk pattern in the middle, or the sort of gradual directional pattern that was expected by most evolutionary biologists prior to the 70s. And you'll see from a paper that Gene published in 2007 and then updated in 2014, there's very little empirical evidence in favor of these directional patterns that are expected by population genetics and what we call the modern synthesis. About half of the data support stasis, a little less than about a third, and then just less than half supports this random walk pattern. So the sort of intra-species adaptive processes don't always translate in a selective framework to the gradual directional changes that we would expect to see. In fact, most of what we see is actually a random walk with no obvious suggestion of directional change or actual stasis. So at least in some taxa, there's considerable support for either stasis or random change, suggesting that what happens within populations of a species is not cumulative across species boundaries. Part of this is because these patterns of stasis are accompanied by these relatively rapid shifts between peaks. It's actually very hard to capture this bit of change within the fossil record. This is an example from stickleback fish in the Miocene of Western North America, in which, because these are lake sediments that you can look at almost year by year layers, Gene and his colleagues were able to collect enough data to actually look at this sort of rapid shift in morphology. Usually we don't see that in the fossil record. We see stasis here and stasis here. Another study by Ueda and colleagues in 2011 in PNAS showed that most change is fairly tightly bounded on time scales less than about 25 million years. So the two purple lines are plus or minus 65% change in body size. The red dot there is the difference in body size between like a Kardashian Chihuahua, or whatever that thing is, and a Great Dane. And that difference in size happened over about 30,000 years. That kind of change does not happen in the absence of human intervention. Kardashians or not Kardashians. Most change up to about 1 million years is really well bounded within this plus or minus 65,000 or 65%. After 1 million years, really after about 25 million years, you see episodic shifts that are much larger. So the waiting time for these shifts is long, but if you wait long enough, they're going to happen. That's a very different pattern of evolutionary change than most evolutionary biologists expected before the late 1970s or 1980s. So most of these evolutionary bursts are relatively modest within an adaptive zone, excuse the extra D. But some of these changes carry species beyond what we think of as the adaptive framework and into a new evolutionary opportunity. Another level of disconnect happens at the level of climatic cycles. This is one of my favorite places in the US. This is the Permian Reef Complex in West Texas. That's a 250 million year old carbonate reef that lay at that time in almost exactly the same latitude that Singapore does today. It's now been exhumed, so we can go crawl all over it and have fun as geologists. This is a cross-section that's sort of actually done straight back from El Capitan Peak here. This shows a prograding carbonate complex, and then the back reef is in green here, and then these sands and carbonates that come down off the reef into the front. The important part of this, because this is all looks like crazy stuff to all of you, I'm sure, is that this wasn't all deposited at one time. Time is moving this way through this diagram, and the timelines go like this. You can sort of see them. They're flat up here, They go down like that, and then they flatten out down here. So another one comes along. And this is a prograding carbonate sequence. And sea level is also going up and down at the same time. So sequence photographers, the people who spend their time doing this, mostly for oil companies, come up with all kinds of great diagrams like this with high stand systems tracks, and nobody actually knows much what they're talking about. But we don't need to, because the key is that these kinds of patterns that we see in the rock record are driven by sea level fluctuations that are ultimately driven by climatic patterns. And there are multiple different cycles of climatic variability. We all know that there are cycles within a year. There are solar cycles over the 1 to 10,000 year, or 10 kiloyear, 10,000 year cycle. And then the important ones for the fossil record are the Milankovitch bands. These are 20,000 today. There are 20,000 year, 40,000 year, and 100,000 year cyclical patterns that drive climatic change. This is what caused or has caused the waxing and waning of glaciers in the northern and southern hemisphere. It turns out that these climatic patterns, which can be picked up on and off for at least the last 400 million years, are responsible for another level of decoupling between the species patterns that happen within the climatic cycles and then across climatic cycles. So when we get a sudden climatic shift, for example, a major extent of glaciation, that will often wipe out, in some regions, a lot of the taxa that were well adapted to the interglacials. And simultaneously, when the glacial cycles end, that also leads to the elimination of taxa that were well adapted to that climatic cycle. But that happens over a larger time span, a longer time span than the ones that I've been talking about. Over an even longer scale are mass extinctions and other biodiversity crises. This is a graph from 1999 from Jack Sikorsky at the University of Chicago. And I'll show you a little bit more updated data in a minute. But this is time along this axis, so going back to the origin of animals a little bit over 500 million years ago. And this is the number of marine genera. So species are accumulated into a genus, the genus Homo for humans, for example, but this is all in the oceans, all in the marine record. And you can see that there's quite a bit of change in the number of genera going up and down here. And through this, paleontologists have identified 5 major mass extinctions. The Cretaceous event that wiped out the dinosaurs, fortunately for all of us, is here about 65, 66 million years ago. And there are several other older events, including this nice event here at the end of the Paleozoic, which almost wiped out everything on the planet. Which, of course, is, I mean, that's a lot more fun than dinosaurs. This is a more recent update of what that pattern looks like. You can still see a number of these major extinction events. And these also impose a major filtering effect on the nature of the marine biota. There are a lot of smaller extinctions. This is 18 different mass extinctions from a colleague of mine, Dick Bombach. But these two, really at the end of the Permian, are fairly fundamental. So the life of the last 600 million years was divided up into the Cambrian fauna, the Paleozoic fauna, and the modern fauna. And this distinction between the Paleozoic and the post-Paleozoic at 250 million years was actually recognized by, you didn't know it was 250 million years, was recognized by John Phillips in 1840. The fossils are so different in the Paleozoic with brachiopods and bryozoans and lots of other things that you've never heard of versus the modern fauna that Phillips actually thought that there was a second origin of life. In fact, he thought there were 3 origins of life. There was, godlike trilobites at the beginning, and then he got bored with trilobites. And then we had the Paleozoic era, and then we had the modern era with separate origins in each case. Modern data has shown that there's a statistical basis for what Phillips recognized. And this Paleozoic fauna essentially gets wiped out at the end of the Permian. It's all of the taxa that didn't survive the EndPermian mass extinction. What we think of as the fauna of oceans today, sea urchins and clams and lobsters and fish, are all the survivors of this event 250 million years ago. So to a first approximation, what you can eat at a restaurant when you go out to dinner tonight, if you're eating seafood, reflects who lived and who died 250 million years ago. That's a completely different level of decoupling from the events that I've told you about before. Now, the remarkable thing about the EndPermian event is how fast it was. This is a sort of a little insight into mass extinctions. This is an ash bed here. This is the Permo-Triassic boundary at a place called Meishan in south of the Yangtze River in China. And because there are zircon crystals within this volcanic ash, we can date this very precisely. We can also look at other places. This is just to give you some sense of the change in the biota from a very diverse biota in the late Permian to almost nothing in the early Triassic at Xinjiang. And that was at Meishan, where I showed you that photograph before. We've also worked for the last 20 years at a place called Panglitan. And relative to what Jeffrey was talking about, when I first went to Panglitan, it was a small village of about 5,000 or 6,000 people. And when I went there in 2016, it is a city of 2.5 million. So I don't know how that fits into your scale there, Jeffrey. But fortunately, this section is still preserved. It's about to get buried under a dam. But we have volcanic ash beds that are shown in red all the way through this rock section. And we're now able to show that this extinction happened in less than 30,000 years. Could have been a lot less than 30,000 years. This is not due to the impact of a bolide or something like that. And it turns out that every mass extinction event that we have been able to reliably determine the duration of happens on that scale of 5 to 10 to 30,000 years. And what that means is that it doesn't matter how you insult the ecosystem. Once they start collapsing, they all collapse in a very similar fashion. And that should give people some pause when they think about the possible effects of biodiversity loss today, because even though the cause of the loss today is different from what happened 251 million years ago, very different than what happened here at the Permian, the way the ecosystem is going to respond is probably very similar. And to those, there are a lot of people who like to talk about the sixth great mass extinction, as though we were going to be able to stop it once it starts. The probability, if you're a good Bayesian statistician, the probability of being able to stop a mass extinction once it starts, once you've realized that this extinction is already going on, is indistinguishable from zero. And that's aside from the fact that coming from a city on the east coast of the United States that happens to supposedly host the federal government, the probability of politicians even knowing what to do is substantially less than zero. Actually, even if they knew what to do, they would do the wrong thing. But But that is actually not where I was going with this. On an even longer time scale, this is we've actually got a paper in press that updates this. This is a look at the oxygen history for the last 4 billion years. And from a chemical standpoint, we have actually lived on, there have been 3 different Earths. There was an Earth with essentially no oxygen in the atmosphere or oceans up until some point after about 2.8, 2.7 billion years ago. The onset of oxygenic photosynthesis eventually leads to an increase in oxygen levels with sulfur-rich and oxygen-poor oceans during a period from about 2 billion years ago until about somewhere between 800 and 500 million years ago, and then a gradual increase to the kind of world that we live in today. So most of the Earth history was in a world that's very different than the world that we live in today. But that's the world in which most of microbial biodiversity has come from. This is the latest Tree of Life, actually published just last week in a paper by Jill Banfield in Cell. People have written long papers about this question, and I don't want to get into it too deeply, except to point out that there is this long ongoing debate about what's driving the kinds of patterns that I've just been describing. And this has been described as the contrast between the Red Queen and the Court Jester. Those of you who've read Alice in Wonderland will remember that the Red Queen is running as fast as she can just to stay in one place. That's the view that has been adopted as the view that biotic interactions and biotic processes are forcing species to evolve and adapt as fast as they can just to survive, much less to actually improve. And this paper by Mike Benton, which summarizes a lot of work over the previous decade or so, suggests that both the Red Queen and the Court Jester, the environmentally driven responses, are operating, but on different temporal scales. So on small geographic scales and short temporal scales, the Red Queen may dominate, but over longer spans of millions of years and larger regions, environmental processes may predominate, although that's certainly remains widely debated. So there is no one rate or time scale of evolution. Evolution operates on many time scales, many of which seem to be, at least based on the empirical data we have, decoupled from each other at a variety of different levels. Let me turn to the last phase of this by talking about what are called slow variables. And this is, the beginning of this is based on work with Jennifer Flack and David Krakauer at SFI. This is something that Jessica Flack. This is something that she's been very interested in for a long time. The idea that coarse graining information reduces environmental uncertainty and increases predictability about the scale. So Jessica studies macaques. She looks at macaque behavioral patterns. And she looks particularly at policing, where the more socially prominent macaques within a group will break up fights with other macaques. And what that does is stabilize the social structure. So there's a particular power structure to these colonies. And even though fights break out, the outcome of the fights is predictable given the nature of the power structures. And this power structure is what she calls a slow variable because it changes much more slowly than the internal squabbles do. And the same thing happens in families, right? You know, the kids squabble and the parents have to break up the squabbles. The same thing happens on longer scales in other biological systems. So in coral reefs, you can have a lot of variability on a local scale within larvae on the substrate or between species within a trophic network. But the long-term stability of the reef structures, which can last for tens to hundreds of thousands of years in some cases, depending on the frequency of changes in sea level are essentially a scaffold within which those shorter-term changes occur. So these long-term or slower variables, like reefs, like forests, as Jeffrey described in his talk, limit the kinds of changes that can occur. So these slow variables coarse-grained data, and they occur actually remarkably widely throughout biology. So cell types turn out to have, not the individual cells, but the type of cell turns out to be stable on the time span of hundreds of millions of years. So we can trace back the history of particular cell types long into the early history of animals. Power structures on a much slower scale, physical structures like reefs or other things that are generated by organisms, beaver dams, also provide a kind of scaffolding within which shorter-term environmental variability plays out. The same thing, of course, happens in human societies, not by the kind of scaling that Jeffrey was talking about, by building institutions, a university, a bank, a firm that lasts from 538, imposes the same kind of stability that the structure of a reef does. I don't have time to go into it, but I just want to point out this one paper that appeared in PNAS last year, or earlier this year, by Nordbottom, which actually looks at the dynamics of modularity and interconnections of networks, in which they suggest that they're able to decompose changes into 3 different levels. So ecologically driven change, evolutionarily driven change, and environmentally driven change, similar to the argument that I've been making here, that there are different levels of change which are decoupled from each other. There are a number of other kinds of slow variables which occur as well. One example that probably is the longest duration type of slow variable in the history of the Earth are biofilms. Many microbial consortia form networks, recurrent networks of interaction that can in fact control the redox chemistry of sediments, so that you have microbes with one type of metabolic behavior at the surface, and then as you go deeper into a sediment or into a biofilm, the chemistry of the biofilm changes and the chemistry of the microbes change to accommodate that. These biofilms, in some cases, produce structures that are called stromatolites that we see in the fossil record that go back at least 3.5 billion years. So these sorts of microbial consortia also have the same kind of properties as other types of slow variables. The type that I'm actually more interested in, however, is the fact that one of the properties that we've seen evolution generate, that was recognized in the last 10 to 15 years, is that there are multiple types of inheritance and multiple types of selection that operate within evolution. Most of those have evolved within the past probably, well, certainly within the last 500 million years. Many of them actually within the last 100 million years. So the variety of different types of evolutionary activity has actually increased in the last 500 to 100 million years, not necessarily within microbes, but with animals and plants and other sorts of things. So ecological inheritance, which includes reefs and other sorts of things that are produced by what's called niche construction, are inherited effects of biological activity that are passed from one generation to another that can impact the fitness of populations. Many of these ecological communities also wind up serving as templates or scaffolding for other kinds of biological activities. the last last sort of thing I want to talk about here is the issue of stability. One of the most poorly understood parts of biology is discussed by this, or mentioned by this quote by Dick Lewington. This is from an SFI bulletin in 2003. He says, the real problem for the evolutionist is not to explain the kinds of organisms that have ever existed. The real problem for the evolutionist is how it is that most kinds of potential and seemingly reasonable organisms have never existed. The biggest problem biologists have, or at least evolutionary biologists, is that organisms are lumpy. And by that, I mean cats are like cats, and dogs are like dogs, and monkeys are like monkeys, and cone shells are like cone shells, and lobsters are like lobsters. But there's not integration between them. The gradual transitions that you expect to see between different groups don't exist. Darwin explained that away as a result of extinction. The intermediates have disappeared. And we spent about 100 years looking for them before it dawned on first Stephen Jay Gould and then some others that the intermediates never existed. The reason the phenotypes of organisms are lumpy is a fundamental property of organisms. It's a fundamental property that we don't completely understand, but one that I think is actually related to time. And let me first tell you something about the pattern, and then tell you why I think it actually is related to the structure of gene regulatory networks. So this is from a paper we published in a book in 2013. These are some of the weird organisms from the Cambrian, priapulid worms, and that's Anomalocaris. This is the neatest thing that ever evolved. It's not a dinosaur. It's, that's Opabinia, which has got 5 eyes, and this claw-like thing in the front. It's about 3 inches long. So 540 million years ago, there's a rapid increase in the number of what are called phyla, the biggest groups, arthropods, mollusks, echinoderms, and classes. And this is what we describe as the Cambrian radiation. But the interesting thing is that once these groups appear, they're essentially stable for the next 500 million years. Once you have arthropods, you produce a lot of different kinds of arthropods, but there's a certain stability to arthropods and to mollusks and to vertebrates and all sorts of other things. And that poses a question for us. What has generated this stability over time? And one explanation for this stability, I think, lies in the structure of gene regulatory networks. Courtesy of my colleague, my late colleague, Eric Davidson at Caltech, this is a Boolean structured gene regulatory network for the development of the purple sea urchin, Strongylus introitus purpuratus. It looks horrible. And I should apologize to you for this diagram, except they're actually kind of cool. So these are all, the backgrounds here are different components of the developing embryo. This is the mesoderm here. Each of these horizontal lines is a gene. And the wiring diagrams tell you which genes activate and repress other kinds of genes. So this, for example, shows you that you've got this gene regulatory network. It has inputs up here and signals coming in, and then it specifies the activity of a bunch of other genes that are downstream. There are a couple of things that are amazing about this. One is that it turns out that parts of this network have been absolutely stable on the scale of 500 million years. These genes are in the same wiring network in starfish and sea urchins, which separated 500 million years ago. It turns out that if you want to make mesoderm in a starfish or a sea urchin, you have to have those genes in that recursively layered network. You can go in and experiment, and they've done this for that entire gene regulatory network. You can go in with a variety of different experimental techniques and knock out each of these genes, and you'll get the endomesoderm if you knock out the genes around these 5, but not if you knock out these 5. So this is the same thing that happens for heart. So this is the gene network that built your heart. That's the gene network for a fly. And the names are different, but in fact, the genes are often homologous between these. So the same gene network is at the core of forming a vertebrate heart as it is the heart of a fly or a lobster. This is some of the properties of these networks. The key point here is that components of these networks appear to have stability on the order of tens to hundreds of millions of years. And that provides a stable core to components of morphology, but allows opportunities around that core. So there are lots of differences, obviously, between flies and humans, except when Jeff Goldblum is in the movie. But those modifications, those evolutionary changes, are confined to other components of these networks. So the networks actually have a hierarchical structure. And if you wanted me to go on for the rest of the afternoon, which you don't, and I'm not going to, I could describe other kinds of structures, such as things called TADs, transcriptionally active domains, within chromosomes. The chromosomes actually also have a structure that allows certain parts of the chromosome to be active at different times than other parts of that. All of these different components of regulatory structures act as different kinds of slow variables that reduce the evolutionary change in some parts of the genome while allowing evolutionary change, particularly in the downstream regions. So much of traditional evolutionary biology has actually, it turns out, been concerned with evolutionary changes of these downstream genes, not the core of these gene regulatory networks. And it's only by looking at the structure of these networks that we can identify the slow and fast variables in evolution. I'll just skip some of the details about this. And I want to end with this quote of Lyell Murchison in 1829, where he talks about no causes whatever have from the earliest time ever acted, but those now acting, and they've acted with the same degrees of energy. Because I think one of the really interesting avenues for future exploration in evolution is to understand how the evolutionary process has itself evolved over time. Evolutionary biologists are sort of curiously Newtonian. They want, because everybody in evolution, however religious they may or may not be, holds Darwin as this sort of, at least a saint, if not a god, and nobody wants to disagree with, or most people don't want to disagree with Darwin. And Darwin was, like Lyell, essentially uniformitarian. He wanted the accumulation of slow, gradual changes over time to produce the whole spectrum of the diversity of life on Earth. And the problem is that once we begin to understand the structure of gene regulatory networks and other parts of development in the genome, we realize that some components of that are much more evolutionarily labile than other components are. And that produces what I've described here as slow variables. It produces differences in the stability of different parts of the organism over, in some cases, hundreds of millions of years of stability for components of phyla classes. But this is something that we're only beginning to understand now. We don't, We only recently have developed the techniques that allow us to look at these patterns of stability and the ability to compare organisms that separated historically hundreds of millions of years ago. So this just summarizes a couple of things that I've said here about the complexity of both understanding evolutionary rates in the fossil record or in deep time, and also in understanding how these may have changed over time and how the sorts of evolutionary changes that are experimentally available to us today may be only a limited fraction of those which are historically important. Thank you.
Thank you for a wonderful talk. So we have now some time for questions. Do you have any questions? Yes.
So going back to your human institutions as being sort of a stabilizing force on change, if you get to a world where we have like social media and all the velocity of change going on, are human institutions really up to that as a stabilizing force at this point? And if so, if not, how do they change?
So two responses. One is that I don't do humans. I'm happy 200 million years ago. And my interest declines exponentially as we get up to today. I mean, that's a really interesting question. I think it goes to the question of whether or not the velocity of interactions through social media will somehow swamp the stability of larger scale institutions. In some cases, that already seems to be happening, but that's. It's also only been, I don't know how old Facebook is, but it's not, by my standards, it's not that old. And it will be interesting to see how people adapt to that and how institutions adapt to that. You already see Facebook and other social media devising ways of labeling fake news as fake news. Of course, we have the tweeter-in-chief labeling everything as fake news, so that imposes some problems. And that is an interesting question. I think it's more for sociologists and anthropologists than a gastropod paleontologist. Brian.
Yeah. I'm wondering if you... Is this on?
Yeah, it's on.
I'm wondering if you have thought of any analogies between your GRNs and computer programs. You must have, because it's a correspondence that's often made. You talked about Boolean networks here. With any particular computer application, that is a program that's written, consists of many modules that often consist of modules, but very often the modules don't change as fast. Well, they don't change as fast as the applications do.
Right.
It might be a quick sort algorithm might be in there that was invented in the 1960s, some other algorithm. These are like the kernel algorithms that don't change. Has anybody thought about that correspondence, or have you?
Ricard Soleil has, actually, who we also know from SFI. And he's actually, in fact, I think he had a paper that's, I guess, in the archive. I don't think it's out yet, that addresses that a little bit. So he's the only person I can think of who's been thinking along those lines. But there's certainly obvious parallels.
So maybe I'm just slow, but what happened to what are you saying happens to the gene regulatory networks in the Cambrian explosion?
Well, I didn't actually say that.
Okay, maybe I missed that.
That's a fascinating story. Part of what's happening in the interval between about 800 million years ago and 500 million years ago is the development of a toolkit that allows you to produce complicated developmental patterning. So one thing that Inaki Ruiz Trujillo at Barcelona has discovered with his group is that there's something called a distal enhancer. So in most gene sequences, you'll have gene sequence, and then there'll be an enhancer that stimulates activity immediately upstream of that. About 700 million years ago, a group immediately, basically at the base of animals, invented a different kind of enhancer that may be thousands of kilobases away from the gene that it actually is stimulating. That allows much more complicated regulatory patterning and the sort of spatial and temporal control of regulation that's required to make plants or animals or something like that. And there's I actually got slides here I can show you later. There's a whole lot of things that are actually invented close to the base of animals that allow a much more complicated pattern of development and regulation. And the argument that Eric Davidson and I have made for the past decade or so is that the first animals basically had a lot of different cell types, but they weren't capable for about 150 million years of making limbs, eyes, guts, things like that. And it's not until these organisms were able to put together the more complicated developmental pathways that you could go from having a variety of cell types to having limbs and all the other things that you find in animals.
It's just an accident that happened.
Well, it's much more complicated than just an accident. There's a lot of environmental changes and ecological changes happening at the same time, including this increase in oxygen. And part of the challenge for those of us who work on the Cambrian is trying to pull apart the contributions of development from ecology from environmental change.
Douglas, you say you don't do humans, but in fact you do. Or at least
not voluntarily.
You drew attention to some parallels between paleobiology and human cultural evolution, etc. Although you didn't go as far as I thought you might go, but Jeffrey did. And I'm interested whenever I talk to, shall we say, people in the traditional humanities and social sciences about these issues, leaving aside economists, most of whom I'd like to leave aside permanently,
Sorry, Brian.
well, not including you. But it seems to me that there's a huge pushback, that the sciences are taking over what should be the role of the humanities and social sciences. Now, my view is somewhat different. We need the approaches of the sciences to help with social issues and institutional issues, more than ever. And I think Jeffrey illustrated this morning. But what is, so I guess I'm asking both of you the question, what is your experience of getting the humanities and social sciences, at least some members of those areas, on side for this large, what Jeffrey's calling a major cultural change without specifics? And when I work at NUS, I don't see very much in the way of collaboration in this sort of area. In fact, there's a huge amount of pushback. You cannot reduce human beings to equations.
Well, so I'll let Jeffrey respond in a minute. Many of my paleontological colleagues, especially in the 80s, would have had exactly the same response. You can't reduce the fossil record to equations. Well, actually, by building a conceptual model, they were doing the same thing, but without numbers, because they didn't have the math skills to do the numbers, right? So I certainly think there are cultural biases against these sorts of modeling things, but when I go to the American Archaeological Association meeting in a couple of weeks in Washington, it's going to be full of young people that are building all sorts of increasingly sophisticated models to look at cultural properties all across the world. So, you know, I think some of that's a generational change. And I certainly think that there is great reason for the humanities and the sciences to talk to each other, and there always has been. I mean, you know, the two cultures thing probably wasn't even true when that book was written. But Jeffrey, do you?
Well, no, I think it's a really important question, and certainly you know, if we're to address some of the big questions and big challenges, we'll need, you know, much more dialogue, if not integration, across the social and more physical, mathematical, physical sciences and the humanities. And it's, you know, it's not easy. And you know, I've reached out some, and I certainly reach out to, in a much more pragmatic way, to people involved in questions of sustainability. I mean, that is on the humanities social side of that. And also, much more practically, for those interested in, you know, the future of cities and specific cities. So it's much more, that's much more to do with practitioners and so on. But I do agree with you, and very strongly. And I think that universities are simply not playing a big enough role in doing this. It's natural, obviously. It's one of the vehicles that we have for fostering such interactions. But society at large, we don't do that very much at all. I mean, on the media, for example, we don't. We do very little of that.
You know, this isn't strictly about social issues and sustainability, but most of my work now focuses on novelty and innovation. And some of the most interesting people that I talk to, most interesting colleagues, are philosophers. And there's a fantastic group of young philosophers of biology that have thought very hard about problems of novelty and innovation in a very different way than a biologist or a paleontologist would. And those conversations and those meetings are incredibly fruitful. Yeah, I think since... I think they are going somewhere, yeah.
Can I just have one thing, but you picked up on something I intimated, at least, was that I do think we do need a major cultural shift, and I would even go as far as almost a revolutionary change. And, you know, I don't know what the forces are at work to make that happen, but there may well be organic forces that will actually force that upon us one way or another. That's what I mean.
Any questions?
Ready for lunch?
Yep. One more question there.
I have a small question. You mentioned that the variables constrain the possibility of environmental changes, but I assume that there are also certain uncertainty about the slow variables, and I want to relate to the last presentation. If we use models to describe certain phenomena, and we have parameters in the model, can we consider the parameters as slow variables, that they are relatively fixed and do not change much, but like under the context of global warming, we still observe slight increase of the parameter value. So
that's a really interesting question. But one of the things that David and Jessica have been doing at SFI is building models that actually have variables with different response rates. But that's, I mean, that's an issue of how you build the model and whether trying to understand how variables with different response rates will respond to the process that you're trying to study. But it's something that can certainly be modeled. Thank you.
Let's thank Douglas Erwin one more time.